viewers. Today I am going to discuss on hypersensitive reactions or hypersensitivity. Hypersensitive reactions are inflammatory reactions which take place during an inappropriate immune response. Hypersensitive reactions were first discovered by Charles Rickett and Paul Portier. They noticed that some individuals overreact in the presence of some antigens, and they coined the term anaphylaxis, the opposite of prophylaxis. Later, different types of hypersensitive reactions were found, and Philip Gell and Robert Coombs, they classified all the hypersensitive reactions into 4 groups. The type 1 hypersensitive reaction or IgE mediated hypersensitivity, type 2 or antibody mediated hypersensitivity, type 3 or immune complex mediated hypersensitivity, and type 4 or delayed type hypersensitivity. The first 3 types of hypersensitivity are dependent on antibody, thus they are part of the humoral immune system. Whereas the 4th type or delayed type hypersensitivity is dependent on T cells, thus it is a part of the cell mediated immune system. Now I will discuss first type 1 or IgE mediated hypersensitivity. Type 1 hypersensitivity is caused in presence of some molecules called allergens, and it is like a normal immune response. The only difference is here only IgE immunoglobulin isotype is generated. Normally, in normal individuals, IgE isotype is generated in response to parasitic infections, but some individuals, they have a hereditary predisposition called atopy, where IgE isotypes are generated in response to common environmental factors which are not antigen or immunogen. Thus, any molecule other than parasites which are able to induce an IgE response is called allergen. Now, allergen or presence of type 1 hypersensitivity is easily detected if a small amount of that molecule is injected intradermally, then immediately a red and swelled area is seen. This is called Whale and Flare reaction. Presence of this area gives a positive result that that particular individual has allergy against that particular allergen. Now, Whale and Flare reaction is caused by the presence of IgE isotype. Like all other immunoglobulin isotypes, IgE is composed of 2 heavy chains and 2 light chains. This region is responsible for binding to the antigen or allergen, and this is the region which is responsible for mediating the biological activity. This region is called the Fc region. And in IgE, sequence of constant region of heavy chain has a sequence called epsilon. And this region can bind to on the surface of mast cell and basophil through receptors. These are the receptors which can bind to the Fc region of IgE isotype, and these regions are present on mast cell and basophil surface. And it has been found that almost 40,000 to 90,000 Fc receptors are present on the surface of mast cell or basophil. Now, mast cells are found in our connective tissue near blood and lymphatic vessels, also under skin, and in skin in 1 millimeter cube area we can see almost 10,000 presence of 10,000 mast cells. And basophils are lymphocytes which circulate in the blood of most of the vertebrates. Now, the cytoplasm of both basophil and mast cell contains a number of membrane bound granules, and the granules are full of pharmacologically active molecules. And these pharmacologically active molecules actually mediate the allergy or type 1 hypersensitivity. Now the primary mediators present in the granules are mainly histamine, heparin, eosinophil chemotactic factor, neutrophil chemotactic factor, etc. And secondary mediators are generated after the target cell activation and during the breakdown of the membrane phospholipids. And secondary mediators include prostaglandin, bradykinin, leukotriene, different cytokines, etc. Now I will tell you the mechanism of type 1 hypersensitivity or allergy. Now what happens, as an allergen enters into the system, it binds to its specific receptor present on a B cell surface. The receptor is called B cell receptor. The B cells are antigen presenting cells. They engulf the allergen, they chop it and they present part of it to their surface via class 2 MHC molecule. Now T helper cells, they recognize those MHC2 and foreign peptide via the receptor present on the T cell. Now after activation or after recognition of the foreign molecule, the T cell and B cell both get activated. B cell continue cell division for several days and produce a clone of B cells. Some of them are converted to plasma cell or antibody secreting cells and some remain as memory cells. Plasma cells secrete IgE isotype immunoglobulin. And once the IgE isotypes are generated, they will bind to the surface of mast cell and basophil through the receptor. Now, if the same allergen enters into the system, then that allergen readily binds to the antigen binding site of the IgE molecule bound to the mast cell basophil. Mast cell basophil with bound IgE molecule is called sensitized mast cell or sensitized basophil cell. So next time an allergen enters, it readily binds to the sensitized mast cell or basophil. Now within 15 seconds of cross-linkage between allergen and IgE molecule, a number of events take place in the cell. Like a number of different protein kinases get activated, membrane phospholipid methylation take place, and eventually what happens, calcium ion channels form on the membrane. And those calcium channels allow the influx of calcium inside the cell. Now the granule membranes also allow the movement of calcium inside and water inside. The granules swell and the swelled granules fuse with the plasma membrane, and all the pharmacologically active mediators present inside the granule, they are released into the blood or in the site where the allergen has entered. Now these pharmacologically active mediators, they now cause inflammation. That means they cause vasodilation, they increase the vascular permeability. So plasma enters into the tissue and many cells like neutrophils, macrophages, all of them enter into the tissue because they sense the presence of a foreign molecule. Now all these molecules, all these cells, neutrophils, macrophages, they are phagocytic in nature and they release a number of different enzymes, reactive oxygen species, reactive nitrogen species, which then try to eliminate the organism or eliminate the foreign molecule. Now normally this mechanism takes place in presence of a parasitic infection. Okay, and reaction stops after the parasite is removed from the system. But in case of allergy, in case of allergic individuals, this reaction takes place in response to many different molecules which are non-pathogenic at all. So the detrimental effects far outweigh the beneficial effects. And that is why this type of reaction is called allergy or type 1 hypersensitivity. Now if this reaction takes place in the blood, if some molecule enters directly into the blood and then type 1 hypersensitivity takes place, then it will lead to the destruction of many different organs and cells, and it will be called systemic anaphylaxis. But if the type 1 hypersensitivity or allergy is restricted to the site of allergen entry, that is if it is localized, then it is called localized anaphylaxis, which is seen in case of asthma, eczema, food allergy, etc. Now, type 1 hypersensitivity can be easily detected if we inject a small amount of that molecule intradermally or subcutaneously, then local mast cells will degranulate very quickly, and swelling and reddened and swelled area will be visible. The whale and flare reaction will take place. And looking at those swelled area, it can be said that that individual has allergy to that particular molecule. And nowadays very easily hypersensitivity can be or allergy can be detected by ELISA, enzyme linked immunosorbent assay. Now I will discuss on type 2 or antibody mediated hypersensitivity. When antibody destroy the red blood cells, it is called type 2 or antibody mediated hypersensitivity. There are three common examples. The first example I am going to tell you is transfusion reactions. There are many proteins and glycoproteins present on the surface of our red blood cell, and they are encoded by different genes, and each gene has different allelic forms. Like we can take the example of ABO blood group system. Some individuals have blood group A, some have B, some have O and AB. Now, A blood group A individual has A antigen on its RBC. B blood group individual has B antigen on its RBC. AB blood group individual has both A and B antigens on its RBC. And O blood group individual has no AB antigen on its RBC. The immune system present in the A blood group individual, when sees B epitopes present on the microorganism surface, the immune system recognizes the B antigen as foreign and mounts an immune response, and thus the A individual, blood group A individual has A antigen on its RBC and anti-B antibody in its serum. Similarly, B blood group individual has B antigen on its RBC and anti-A anti-serum or anti-A antibody in its serum. So, if A individual is transfused with blood from B individual, then what will happen? The B antibodies present in A blood group individual will bind to the B antigens present on the RBC of the B blood group, and readily it will lyse the RBC, and it will, at this mechanism, this reaction is called type 2 hypersensitivity, which is seen in transfusion reactions. Another example of type 2 hypersensitivity is hemolytic disease of the newborn. A common example of hemolytic disease of newborn is erythroblastosis fetalis. What happens? We know that on our RBC there is a protein called Rh or rhesus factor. Now, if the mother is Rh negative but the fetus is Rh positive due to the father, then mother's immune system will recognize Rh protein present in the fetus as foreign. Now, what happens during the first pregnancy? It creates no problem, but during delivery some blood from the fetus enters into the mother's circulation system, and mother's immune system recognizes the Rh proteins present on the fetal red blood cells as foreign and mounts an immune response, and plasma cells and memory cells are generated. The plasma cells secrete IgM antibodies which readily clears the fetal red blood cells, and memory cells are kept in the circulation. Now, during the second pregnancy, if there is any blood contact between the fetus and mother, normally there is no blood contact due to the presence of the placenta, but if by some due to some damage there is blood contact between the fetus and the mother, and fetal blood enters into the mother, then the memory cells generated during the first pregnancy or first delivery, they will be activated, and this time memory cells will secrete IgG antibodies. IgM could not cross the placenta, but IgG isotype can cross the placenta readily. They will cross the placenta, they will go to the fetus and they will destroy all the red blood cells, and it will create anemia. Mild to severe anemia can be observed in this case. But erythroblastosis fetalis can be prevented by administration of anti-Rh antibodies after the first delivery. If these antibodies are delivered within 24 to 48 hours after the delivery, then these antibodies, preformed antibodies, will readily destroy the fetal red blood cells, and it will not give the mother's immune system to see them and get activated. Now, a third type of type 2 hypersensitive reaction is called drug-induced hemolytic anemia. Some drugs like penicillin, cephalosporin, streptomycin, they can bind to some proteins present on RBC surface non-specifically, and after binding they form a large complex and antibodies can recognize them. They act as hapten carrier conjugate. Now, antibodies can bind to those drug protein complex and readily destroy the RBC by complement mediated lysis. Now, I will discuss on type 3 or immune complex mediated hypersensitivity. When an antigen enters into our system, if specific antibodies are present, readily they bind, and then phagocytic cells remove those immune complexes from the circulation. But if the immune complexes are not removed properly, then they can be deposited in many places, like the wall of the blood vessel, synovial membrane of the joints. Also, they can be deposited in kidney, also in brain, and in many other places. Now, once an immune complex is deposited at a place, then it will activate the complement system, which is composed of a group of proteins present in our serum, and some of the complement split products, they act as chemotactic factors. That means they will call some cells like neutrophils, macrophages from blood to enter into that tissue. So, in case of an immune complex deposited in a tissue, many cells like neutrophils, macrophages will move to the tissue and they will try to eliminate that immune complex. And in doing so, they will secrete a number of enzymes, reactive oxygen species, reactive nitrogen species. And all these enzymes and these reactive species, they will kill or they will try to eliminate the complex, but at the same time they will damage the normal tissue. This is the basis of type 3 hypersensitivity or immune complex mediated hypersensitivity. Now, if the immune complex is formed under the skin intradermally or subcutaneously, that is just at the site of the antigen entry, then what happens locally, there will be a swelling and a swelled area can be seen. This is called Arthur's reaction. And if this reaction immune complex is formed in blood, then they can be deposited in various different organs and they will start a number of disorders in the immune system, which will ultimately lead to a number of symptoms collectively called serum sickness. And sometimes the consequence may be fatal. Now, I will tell you on the fourth type of hypersensitivity, that is type 4 hypersensitivity or delayed type hypersensitivity. Now, delayed type hypersensitivity has two phases, sensitization phase and effector phase. Now, this type of hypersensitivity is mainly seen in the presence of intracellular bacteria or virus. That means the bacteria, viruses which reside inside the cell where antibodies could not act or reach. That at that time delayed type hypersensitivity is seen. What happens if an intracellular organism is growing inside the cell? Then the antigen presenting cell will present a part of it on its surface and Th cell will be activated. seeing at the class 2 MHC molecule and the foreign peptide. Now, when a T cell, T helper cell recognizes class 2 MHC plus foreign peptide, it gets activated, it will start division, and some of the cells will be converted to effector cells, some will be converted to memory cell. And this time the T cells will be converted to Th1 subtype. Now, next time the same virus or bacterium enters into the system, and the sensitized Th1 cell recognizes the same pathogen or same foreign peptide presented by class 2 MHC on the surface of the antigen presenting cell or on the macrophage, the sensitized Th1 cell will secrete a number of cytokines. Those cytokines will activate the resting macrophage. The macrophage will be activated and it will secrete a number of cytokines. And all these cytokines will together try to eliminate the intracellular organism, but in doing so they will call more cells to that site of infection. So, what happens in a fully developed delayed type hypersensitivity? It is seen that a giant ball like structure has been produced where 5% of the cells are of Th1 cells and 95% cells are of macrophages. Since macrophages rush to that area to kill the intracellular organism. And this is called delayed type hypersensitivity because the peak response is attained at around 48 to 72 hours starting from the effector function. Okay, that is why it is called delayed type hypersensitivity. Now, delayed type hypersensitivity can be detected also very simply if the molecule is injected intradermally or subcutaneously, and if sensitized Th1 cells are present under the skin, then readily they will secrete cytokine and they will call the macrophages. Macrophages will accumulate to that area and they will form edema, a swelling or edema like structure. And a very common delayed type hypersensitivity test is called tuberculin skin test or Mantoux test, where a protein derivative derived from the cell wall of Mycobacterium tuberculosis is injected under the skin. If that individual is sensitive to Mycobacterium, then he will have sensitized Th1 cells. Those Th1 cells will rush to that area and macrophages will come and a swelling will occur. Measuring the diameter of the swelling, it can be said that whether the Mantoux test is positive or negative. DTH response, delayed type hypersensitivity response, can be observed in the presence of number of molecules like hair dye or pollen, grain, etc. One example, important example, is through poison oak. From this oak tree, a molecule called pentadecacatechol is produced. This molecule binds to the proteins present on the skin, and this protein catechol molecule, this conjugate is readily engulfed by the macrophages. They present it to the T cell and type 4 hypersensitivity occurs. So, today we learned the word hypersensitivity means hyper activation of the immune system, but actually it is not hyper at all the time. It is actually inappropriate immune response. We also study different types of hypersensitivity. There are 4 types of hypersensitivity. 3 of them are mediated by antibody and the 4th type is mediated by cell, T helper cell. We also studied the mechanism of action of each type of hypersensitivity, and we can avoid the causes of the hypersensitive reactions. Like in case of allergy, if it is a systemic one, then we have to administer epinephrine, that is the choice of medicine. But in case of localized anaphylaxis, or in case of localized type 1 hypersensitivity, if it is from food or from asthma, from dust allergy, then it is better to avoid those things. And in case of type 3 hypersensitivity, type 4 hypersensitivity, if it is possible to avoid those allergens or causes of hypersensitive reactions, then it is better. Thank you.